Clock is ticking.
Hello everyone. I'm going to present this lecture's article titled Dendritic Asymmetry Cannot Account for Directional Responses of Neurons in Visual Cortex by the group including Idam Shikef. The motivation for the study. Okay, no. Sorry. Okay. The motivation for the study. Uh, there are several models proposed for the directionality of the neurons. Few of them we also discussed in class. One of the correlation models based on temporal summation or multiplication of excitatory input in the preferred direction of motion in flight, and one model for the rabbit's retina uses the fact the inhibition of excitation in the non-preferred direction of motion to achieve directionality. And these are the models which are based on the spatio-temporal asymmetry in input for the two direction of motion. In this paper, they are going to use the Livingston hypothesis, which says that excitatory synapses are activated sequentially along asymmetric basal dendrites of large solitary pyramidal cells of Maynard. In simple words, this hypothesis says that in a long basal dendrite, if an excitatory region encounters first, then the cell will fire, and if inhibitory region encounters first, then the cell will not fire. Okay, so the goal of study is to test this Livingston hypothesis experimentally in the cat by recording from directional neurons. And these are the methods. They get in vivo intracellular recordings and extracellular recordings for the mapping of receptive fields. And the procedure follow, like from mapping, they fix the sections and did microscopy and then make 3D neuron reconstruction and then project that 3D neuron reconstruction on 2D surface. They group the neurons in three categories: the non-directional and the directional preference and directional selective. The non-directional one, which respond for backward and forward motion, backward and the forward motion both. Directional preference, which are biased for one direction of motion, and directional selective, which are exclusively firing for one direction. Okay. Now in this, they wanted to calculate the dendritic bias or the asymmetry in the dendrite. So this is the projection of that 3D reconstruction of neuron on 2D surface. In this, the black dot in between is the soma, and L is the distance from the soma to the most distal dendritic tip. That cone is of 30-degree angle, and that shaded area is 1/3 of L, and phi is the angle between the mid of the section and the horizontal axis, and R is the sum of the dendrites within that gray shaded region. And so R and phi, they are the measure for the dendritic bias, and they did this kind of study for pyramidal neurons in monkeys and cats, and this is the distribution of R that they got by each degree of phi, and this is the direction of preferred motion. And phi max is, they named it as dendritic bias angle.
I have a question.
Yes?
So I don't think I understood. This is the morphology we're seeing here?
Yes.
And what do you mean, what is this, like the preferred direction?
They presented some stimulus, and there is one preferred direction for each of the, like this cell fire for this direction. These are the direction selective cells, so they fire for some preferred direction. And this arrow shows that this is the direction of preferred, this is the preferred direction.
Of stimulus is
but they made this thing from the fixed section of like they fixed the section, and you you can like if you want to turn it the section and then make the 3D projection, it's just like you are manipulating your result like you want to be.
So these are, where are these neurons?
These are the neurons in visual cortex,
Yeah.
and they cut the section and they did the microscopy and made the 3D reconstruction of those neurons, and then they projected them on 2D surface.
Okay, I'm still not sure that I understand why there should be a relationship between the orientation of because the neuron is just the orientation of the neuron is arbitrary compared to the stimulus. I don't know. Maybe if you go on, it will become more clear.
I can. Yeah, this is the thing. This is the receptive field direction frequency of the neuron. Okay. So, okay, this is how they calculated the dendritic bias in all the neurons. And then they did some kind of analysis, which includes the angular difference. Angular difference is the difference between the dendritic bias angle phi and the preferred direction of motion, that arrow, the angle difference between the phi and that arrow. And figure A is the angular difference for different cell types, and figure B is the frequency distribution of figure A. And the Livingston hypothesis said that the optimal difference should be 180 degrees, but they found that that almost none of the cell is giving 180 degrees, and all the angular difference covers the entire range. Now, the second analytic parameter was to find out the difference, which is the asymmetry of the dendritic tree, in which they calculated the difference between the R's of opposite segments. Like one of the R is there and one of the R of opposite segments, and these are the different cell types. The filled dots are the R's which are along the angle phi, and the open squares are the R's which are along the preferred direction, that arrow. And you see almost all of the squares are clustering around 0, which says that the preferred direction of the receptive field doesn't have this dendritic bias feature. There are two dots which are showing negative, and these are the exceptions, which are in according with the Livingston hypothesis. And this is the polar plot from this figure, and the radius along this plot shows the degree of asymmetry, and the angle is the difference between phi and the preferred direction. And most of the neurons are not showing the asymmetry. Only a few neurons are showing this, and if they are aligned along a good axis, or what should I say?
Sorry, once again.
Yes.
Operation. I think you need to understand. Can you explain once more what is phi, what's the asymmetry, what we would have expected, and why this is like A is not what make 180 an optimal for what?
Okay, so if this is the angle phi, and this is the preferred direction, and the difference between these angles
What's the
is
first? This is what I don't
okay,
understand. What's the first angle?
this thing from the horizontal to this.
What is this?
This is the distribution of R max. Like this is the neuron. At each 1 degree, they calculated R max.
Which is the maximal value of what?
Of R.
Fundamental. So they take a specific section on the 2D projection,
Yeah.
and then just measure the length, the sum of the length.
Yeah. Not the overall length. They just took 1/3 from the top, so that they can exclude the bias from the small dendrites. And this is the distribution at like each 1 degree step. They calculated this R, and this is the distribution of that. And they found that R has maximum value here, and so they named it as phi max.
Which means, so the gray area is the histogram
Yes.
of the sums of the length.
Yeah.
So this is just like density.
Yeah, and it is like superimposing one histogram
dendrite, what we would have expect?
Yeah, if this is like if the difference between this, if this arrow would be here, so the difference is like 180 degrees. Then they are like asymmetric dendrites,
Okay.
and we can say that this asymmetry is the reason for the directionality.
What is asymmetry between the density and the?
Uh, Density, like this is the direction for the receptive field. Okay, receptive. Each neuron has one receptive field. Okay, maybe I no, I don't have anything
No,
else.
it's okay. Go on.
Okay, this is how they are calculating the dendritic bias. This is the property of the neuron. This is not that they are manipulating, and this is what they are calculating. So it may be here, 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 here. But in all the neurons, they found that only 2 or 3 neurons are like that. That this arrow lies just opposite to that of phi max.
And the arrow is the preferred direction of the
Yeah,
receptive
receptive
field
field.
outside world, not related to the
Yeah,
tensor.
not related to the analysis.
How did they measure this black arrow? Electrophysiology?
Yeah, from the
From the
recording.
dendrite?
Yeah.
Okay.
No, from the neuron.
So, okay.
Like you do some electrophysiology and your stimulus is like this direction or this direction, then neuron fire, then you
From
assign
the
okay.
stomach?
Yeah, they did it like by hand.
I think what still confuses us, three of us, what does it mean? It may be not so important, and then please go on. What does it mean, like, if there's a great density of matter, of dendrite, up there,
So,
and
so
what does it mean to, like, up there is kind of, like, arbitrary in related to the preferred down direction, which is, like, you stabilize the head of the animal, the preferred direction is down in the outside world. But in this, why should there be any relation between the area in the brain, like up, like in here there's a great density, and that the preferred direction is down?
Maybe you are considering two different things. So,
Maybe.
yeah, so the neuron they are considering are maynard neurons, which are very sparse and they are mostly found in most of the layers of auditory, oh sorry, visual cortex. And so the hypothesis says that there are lots of synapses along the long dendrites which manipulate the directionality. Like if the excitatory synapse encounters first, then it will fire. If inhibitory encounters first, it will not fire. And if there is any kind of linear summation, then only the voltage threshold for spiking determines the directionality of the neuron. So this is the hypothesis, and in this study, they are going to see if this hypothesis is true for all the neurons, if it generalizes or not.
Great, thanks.
Okay, so until now it's not.
Okay, Kavini, you have 5 minutes left, so maybe
Okay,
we can.
so now, so this was not the generalized case. They wanted to see it for all the neurons, but the Maynard neurons are not so good to study for all the frequency. So what they used for their problem, they did the computer simulation. They modeled the Maynard neuron, and the assumption was that all the retinotopic inputs are on one long vessel dendrites. They use 2000 ampere excitatory synapses which are uniformly spaced, and they activated them in a temporal sequence with different sweep duration. And they studied D2P, like distal tip to the soma and proximal dendrites, and P2D is the null direction from the proximal dendrites and the distal tip. And this is the graph. They measure the voltage transient at the soma with the variation in sweep duration, and they found that the optimal sweep is at 10 milliseconds, which was in accordance with the Livingston study. And 0 is the sweep when all the 2000 NPA receptors are activated simultaneously. And in P2D direction, the maxima also occurs at 10 milliseconds, and it decreases as we increase the sweep duration. Intuitively, sweep duration is the propagation velocity of the signal from soma, of the EPSP signal from soma to like the end of the connection. I think I'm very fast in this. Okay, then they wanted to check if their model is good or not. They compared it with the infinitely long cylindrical cable, and we studied in class that for infinitely long cylindrical cable, number is almost equal to 2.8 lambda by tau m. So, and the other parameters like space constant, tau, and optimal velocity, they are also in according with the theoretical value. So they can, so they decided, okay, their model is good, and let's proceed with further studies. Later on, they added some. They wanted to check what if they add some excitatory synapse on the opposite dendrite, not on the basal dendrite. And in these are the curves for one dendrite. That means only one. All the synapses are on the one long basal dendrite, and these are the curves for two dendrites. That means some 2000 synapses on the long dendrite and 600 synapses on the short opposite dendrite. And they found that the effectiveness of the delay lines strongly depends on the membrane time constant. You see, when we increase the time constant from 5 to 50, the velocity, voltage peak occurs at some velocity, which is the optimal sweep duration for EPA speed, and it decreases. So it's not the property of the length of the dendrites or dendritic asymmetry which accounts for directionality. It is the property of the membrane time constant and other membrane parameter which affects the propagation speed and hence manipulate the directionality of the neuron. Uh, and with these two dendritic things, they found that the velocity profile, initially they have some kind of peak, but with two dendrites it's kind of saturate. And in such cases, there is a loss of directionality on the basis of this hypothesis because there is no symmetry. But in cats, most of the neurons are like short dendritic neurons and they do have directionality. So conclusion, there is no correlation between the dendritic bias, like dendritic asymmetry and the directionality. It is not a sufficient condition for directionality. So, and Livingston hypothesis may apply to some of the neurons, not all. The temporal activation of dendritic tree, it has a little contribution for the computation of directionality, not overall dependency, and this computation must rely on the network mechanism. Thank you. Okay.
Oh, we need to discuss, huh?
Yeah.
Not a lot. Everybody understands if we give you an exam now, you should be able to answer the... Oh, Kamini, can you go back a step?
This one?
Yes, I'm trying to understand what is the connection between this model and what we saw before.
Uh,
Before you made an empirical observation.
Okay.
Right?
This one.
Yes.
Okay. This is from the experimental study. Like they did some
No, this
recording
I
and then
understand. What is the computational? How does the computational part?
is different. No, the computational part is same for the model neuron. Like this analysis is same for. I'm not getting the question maybe.
Yeah.
Yeah.
So now this model is based on, they took the other neurons, the morphology of the neurons, modeled them to get the receptive fields. I'm a little bit lost on what did they do.
Okay. So they wanted to check the generalized case of Livingston hypothesis. For that, they took the best asymmetric possible model of Maynard neuron. Like
From the real data?
they made a computational, like they modeled one neuron from the real, like they wanted it to be like similar to the real Maynard neuron.
Yeah.
It's like I'm making one pyramidal neuron computation. I'm doing some program for.
So they're modeling the neurons they recorded from.
Yeah.
Both that it will fit the anatomy and the physiology of the cell.
Yes.
Okay.
And this is how they are checking it. Like they found the velocity to be 75 mm per second, and in the experimental data it was 77 mm per second. And the length constant, yeah, the length constant is according with all this. Yeah, this analysis they did it for to check if their model is in according with the original physiology of the
They
neuron.
can fiddle with the parameters until the model fits the recorded neuron.
Yeah.
Okay.
And yes, this is what they did.
But
and they are saying like in that model everything was based on the structure of dendrite only, like the length of the dendrite. But it is not only the length of the dendrite. The sweep velocity, which is like less here and high here, it depends on the synapse, on the membrane properties. Because if you have a longer time constant, then the signal can move slowly, and then to build some potential at the soma. But if you have a small window, then the signal have to move fast to build up some kind of potential at soma. No? Maybe I don't know.
Okay, more question.
I-
Everything is crystal clear? Ohad, clear to you?
Do we have time?
Yeah, we have 5 minutes for discussion.
Oh, okay. Can you go back to the
Mhm.
presentation?
Okay, you ask question, I can even if yes.
It was just a little bit too quickly for me. The
Mhm.
last figure or the one,
Sorry. I don't know. Okay. What is the question about? About the last figure.
you've shown us like the saturation of two dendrites.
Mhm.
Can you elaborate about it? Here again, exactly this one.
Okay.
So the difference between the two figures is
Mhm.
tau. What
Yeah,
does
the
it mean
difference
actually, tau?
between just the tau.
What would be changed between the two models?
Uh, the model is same. They just change the membrane, like the tau, the time constant of membrane, from 5 to 50 to check what is the impact on the voltage peak. This voltage peak is the voltage that they measured from the soma, and this is like clear that they are plotting voltage peak as a function of velocity.
And how does this relate to directionality?
Because directionality is okay. So as the hypothesis says that something is moving on a dendrite, and if excitatory or inhibitory synapses are encountered, then they modulate the function of this neuron.
So the movement is of the signal,
From
the
the
electrical signal in
yeah
the neuron, not the movement in the outside world?
no.
Okay.
This is the signal in the neuron which is moving,
Like the
and yeah, and
electrical
the
movement?
hypothesis said that the inhibitory and the excitatory synapses, the connection on the dendrite, which causes the dendritic asymmetry, is responsible for the directionality. This is not the property of the neuron or of the network which is determining the directionality. This is just the morphology or the structure of the dendrite.
So they keep the morphology as it is between these two experiments,
Yeah,
and they only change
change the time constant.
what will it mean? That
The membrane, because in
if I have
every
two neurons
yeah.
in which tau is greater, tau is now 50
Mhm.
milliseconds, what will it mean to the
That
system?
it has more, the membrane time constant is more, and the signal have more time to cross the membrane. And if I'm giving the slow signal, it can. If I'm giving the slow signal, it can have. It do have some time to reach the soma. There is no like
Okay.
loss of signal with
Okay.
more.
So
And this is just, oh, I'm sorry.
No, go
This
ahead.
is a parameter like in Hodgkin-Huxley model we have 1 by tau and dv by dt. So this is I think that kind of parameter only.
so just right. So back to this,
Mhm.
increase tau, and then we see that what happens.
So if you increase tau, you see the tip. Uh, okay, with less time constant, the tip is like here. It's almost like 70 something the velocity, 70 millimeter per second. And if I increase the tau, and it comes to be like 24 millimeter per second.
Does it have any functional importance, or it's just a number?
It's just saying that in actual system it does happen. So they are saying that their model is capturing the features have one peak now. It is like saturating. Okay, and this is like a high pass filter kind of structure. And with such situation, the D to T, and with such situation there is a complete symmetry. Like if I consider the small dendrites with the long dendrites, then the structure on the basis of the analysis is said to be a symmetric structure. Okay, and the hypothesis says that with symmetry there is no directionality. But in cat visual cortex there are many neurons which have small dendrites, and they are directional neurons. So the hypothesis is not correct.
So then
Oh,
the take
so last question and then we move on.
home message is
That the property, the basic property of neurons like membrane constant and other features of the neuron are the important things to study for directionality, not just the structure of dendrite, if it is long or short. Thank you. Hmm?
Okay, so who is up? Name?
Adi.
Adi?
Yes.
Adi, my watch will ring at 20 minutes, which means you have 5 more minutes to completion and then 5 more for discussion, and then we take a break.
Okay.
Okay?
Do they start once I connect everything or
Yes,
now?
I will start once you tell me to start.
Good.
Take your time.
We have this thing. Oh. Something happened. It's not happening. Okay. So I think I'm ready now. It's working. But I'll take my time as well. Uh, okay. So I'll present the article Computational Subunits in Thin Dendrites of Pyramidal Cells, which was written by Polsky, Mel, and Schuller, published in Nature Neuroscience in 2004. And you'll see that despite the year, it has really cool methods in it, and also conclusions, I hope. Okay, so just a short background before we dive into the actual experiment. So the main thing this article will try and show us is how does the pyramidal neuron do math. So first of all, the pyramidal neuron, just so you all are with me on considering the terms. So the pyramidal neuron, I just lost the thing. The pyramidal neuron has the soma over here. Usually it's like a triangular shape surrounded by basal dendrites. And there's the apical dendrites in the apical duct, the famous tree-like structure. And in this study, they will look at the pyramidal cells in layer 5 of a specific rat chain. And when I talk about mass, I actually mean what does this pyramidal neuron do? It gets a lot of excitatory inputs in this case. And in this particular article, it's all sub-threshold inputs. And they will ask, what does it actually do in order to take these inputs and produce some kind of output? So this is what I mean by math.
So the math is the input-output function of the neuron.
Yeah, how it adds inputs. And so just like one of the main theories that they talked about that existed at that time. I'm not sure if up to now someone uses it. Is global summation. Global summation basically means that the cell body doesn't really care where it gets its inputs from. They can be from here, and as long as they're weighted the same way, he will produce the same output. It doesn't really matter. And this kind of addition can be linear, meaning just 1 plus 1 equals 2. It can be sub or superlinear. Sub is 1 plus 1 is less than 2, and super is 1 plus 1 is more than 2. And these can theoretically be combined into a sigmoidal addition function. So this is the kind of model that existed when actual studies were able to model. However, they will try and show why this kind of model is not good, and they will propose a 2-layer model as a better explanation for the kind of summation that occurs at the soma. So just like as a brief, promo for what you're going to hear, these kind of models have completely different predictions as to what the neuron is supposed to do. While in the global summation, I would expect that giving an input here and there, it doesn't really matter. While in the 2-layer model, I would assume that if I give the inputs to one compartment, which will be clear later what I mean by that, I expect a completely different output than when I give it like to a compartment here and a compartment here. I will get something else. So this will be clear, I hope, later on in the talk. Okay, so this is basically what they did throughout the experiment. What they tried to do is to activate specific points on the dendrites and see the somatic voltage when they stimulate it. So now in detail, what you can see over here is a whole cell patch clamp, which they use in order to clamp the soma. And also they injected calcium indicator in order for them to be able to see what they're doing. You can see that inside the pipette there is this calcium indicator as well. And once they injected it, they could see the dendrites, which are really tiny, as we all know. And then they were able to zoom in on a specific segment, as you can see here, and using confocal microscopy. And then what they did is they placed 2 stimulating electrodes, extracellular ones, and they gave small pulses. So first of all, they gave a pulse using like this blue electrode. And what you can see here, it's a lot of graphs, so bear with me. Over here you can see the traces that they saw at the soma. Okay, this is what they were able to measure, and you can see that the blue trace, which is what was the cause of the blue stimulating electrode, and the red trace are basically, they look quite the same at the soma, though they're from different places. But what's more important here is that due to the fact that they had the calcium indicator within the dendrite, they could follow the calcium transient along the dendrite. And you can see that, for example, over here, when we're close to the red stimulating electrode, we have a really large calcium transient. And as we move further and further, it's barely non-existent. And the opposite is true for the blue electrode. So over here we have a large transient. As we move along, it gets smaller and smaller.
Can you just explain again, where is the red and where is the blue, and what's the relationship between that and the graph?
Okay, this is the red stimulating electrode. This is the blue one. And they stimulated, the first time they just stimulated the blue, and they looked.
But where are they on the neuron? Is one at the basal and one's at the...
No. This is just this kind of, specifically in this case, it's this part of the dendrite. Okay? So why is all of this important? Because it was really important for them to prove that they're actually stimulating what they're saying they're stimulating. Since they're going to try and show us what happens when we stimulate this tiny section in comparison to this tiny section, it's really important that we're convinced that they're actually doing that and not just stimulating an entire area of the dendrite.
Just to make clear, the mid-left traces are from the soma. All the rest are imaging.
This is the soma, and this is the delta F over F calcium. Okay, so this is why it was really important. They also have some controls they do. They look at other dendrites, and they see that over there aren't transients that can be associated with the stimulus they're giving, as well as using NMDA blockers, which basically abolish calcium transients, to show that this is actually relevant, what they're doing. Okay, so once we're convinced, hopefully, we can go into the actual question of the article, which is the comparison between within branch and between branch summation. So within branch is the other name for within the compartment, whilst the between branch is different compartments. So now, hopefully, it will be less abstract. What you can see here is the basic conditions they use. The within branch condition consists of two stimulating electrodes placed in close proximity on the same branch, as the name indicates, and the somatic voltage will be measured at the soma. And over here is the between branches condition, where, as you can see, the two stimulating electrodes are placed on two different branches. And again, the voltage will be measured at the soma. So now they can start giving inputs to the cell and see what's going on. So here we see what happens when there are weak inputs received to the cell. And what you can see in black are the two separate traces. First they just stimulated A, measured what happened at the soma, the APSP. Then they stimulated B, so what happened. And in blue is the arithmetic sum, which is basically had the soma added the inputs in a linear fashion. 1 plus 1 is 2. And you can see that this is pretty much the case. The red trace is what was actually measured at the soma. And it's true both for within branch and between branch, in both conditions. So now it's getting a bit more interesting. When they gave larger inputs, you can see that in the between branch condition, we get still a linear summation at the soma. Nothing interesting. Or maybe it is interesting, but not different. But over here, in the within branch condition, you can see that in blue is the arithmetic sum, and in red is what was actually measured, meaning we got a much higher, much bigger change in the somatic voltage, indicating a superlinear summation. And even further, they gave really strong inputs, and now you can see that in this cell, in this condition, there is now the sum actually measured at the soma is smaller than what you would expect had it just added the inputs, indicating sublinear summation. And over here, we can see in the between branch condition that there is slight superlinearity, very small, but still it's no longer linear. So just to sum up what I told you, we have this graph with a lot of dots. But so first of all, the x-axis is the expected peak, which is just a linear summation hypothesis, and the y-axis is what was actually measured, and along the diagonal is of course the linear, what if it is indeed linear. And you can see in green diamonds, hopefully you could all say it yourself, this is the between branches that they measured using different stimulus intensities. And the different colored dots are basically different conditions in which they tested the within branch hypothesis. And you can see that given very small inputs, it's basically linear. Then after a certain threshold, you get the superlinearity, as you could see in this graph. And finally, when the input is really, really large, you get sublinear summation. I will not get into exactly what the color code is unless you ask me later, so there's a question for you if you're interested. And this, again, something else I will not talk about. So now to define the within branch compartment, they tried and look at how big is it. I mean, we're talking about a compartment, but we still have to make more specific. So what they did here is they placed one reference electrode at a constant place, and they had the second electrode and moved it along the dendrite branch. And what you can see here is, again, the same axis as before. This is the expected peak, like the linear summation you would expect, and this was is what was actually measured. So you can see that at 20 microns, when the distance is quite small, there is this, above a certain threshold, you see this superlinearity, as was seen before. However, when the distance is really large, at 200 microns, it becomes basically linear, as if it's from totally different compartments. What I would expect had I stimulated the cell at completely different branches. And in the middle, they chose to show the 60 micron distance, and it seems as if there's slight superlinearity in this range. In the paper itself, they do mention that they tested a different length, like this mid-range length, and they report that sometimes they do see the superlinearity, but not necessarily. So this is kind of an indication that the compartment is not really this strict, bounded part of the dendrite, but rather more fluid, like smudgy edges of the compartment. And
Question?
yes?
Did they mention anything about the branching pattern along this 200 micron? Because I would expect in 20 micrometer there won't be any branching on the middle.
Amazing question. Because what they do now is they showed us here that the 20 microns is a really good length for us, and now they do a really systematic verification of what happens if I take these branches, which are, there's this branching point over here, and they're about 20 microns apart, I think, maybe a little more, I don't remember the exact number, and you can see that still, even when the distance between them is quite close, you still see this linear phenomena in which the soma basically adds up the different inputs, unless you give a really high stimulus intensity, and then there is this kind of superlinearity which they talked about before. Okay? And
Right, so in this case, you don't get a superlinearity when you do a normal.
in this case,
Yeah.
yeah, this is exactly their point. They're saying the compartment has a specific length, as they showed here, 20 microns, sort of, like plus minus. And what they showed us is that it's not just a matter of length, it's a matter of branching points as well. If you have a branching point, you can have really close branches over here, and still you will not get the superlinearity that we see very nicely here. Yes?
Can you go to the next slide?
The one that I'm not talking about? Or
Yeah,
this?
yeah. So here when they show the superlinearity,
Yeah.
what is the length of that
What-
they use? Yeah.
over here, well, that's kind of, yeah, they don't really specify it. They say it's 30 to 50 microns distance.
And by chance they guess that there is no branching in
I'm not
in
sure I
this system?
understand what you're talking. Are you talking about the between branch or the within branch?
Here the branches were the between was very far away.
Here, the between branches was sometimes close and sometimes not.
Okay.
And I
But
think that's...
they're branching off of the soma, though. That's the difference between this and the
No,
other
this
one.
is actually a
Right.
simplified kind of drawing. I think they measured it at around 100 microns away from the soma in both cases because they had like problems with visualizing.
Right, but the difference between A and B in this
Yeah.
case is that A is on one separate branch from the soma.
Yeah.
But in the next slide that you showed, so they still come off of one branch of the soma, but then it splits into two.
Yeah. Okay, and I think in the previous one it's not necessarily the case. It might be, but it's not necessarily. Okay? And so I wonder if I have time. Okay. So I think I'll go back to something I skipped before, which is the nonlinear explanation and some kind of a beginning, an initial explanation of the mechanisms that might underlie it. So what you can see here is what I didn't actually tell you is how they stimulated. I had two stimulating electrodes, and I didn't tell you whether they stimulated, they used simultaneous stimulation or one after the other. And what you can see here is that they tested this, and they showed that whether you do it at the same time, simultaneously, or if you do it one after the other at a 50 hertz timing, then you get pretty much the same thing. They tested it using intensities that are good for superlinearity. And what you can see here is that qualitatively, they're pretty much the same. However, in black here, if you use a pair of EPSPs, meaning it's one after the other, the threshold for superlinearity is lower. You see this curve? It occurs earlier than if you do it just like simultaneously. And what's I think more interesting maybe is that when they put 85, which is an MDMA receptor blocker, an antagonist, you can see that the superlinearity basically diminishes. You see now that the blue and the red, which are what we expected and what was actually measured, are pretty much the same, meaning the summation is now linear. And this is summed up over here, where you can see the same curve, where they use the paired pulse. And in blue is when they use the same cell, but they put in the extracellular fluid the 85, the inhibitor, and the antagonist. And you can see that it basically becomes linear when you put in that substance. Okay, so a final graph before we summarize. The last thing they wanted to do, once we have our compartment, we know how large it is in terms of distance, but how can, temporarily, what do we need, what is the time scale in which it knows how to calculate? So over here, what they did is they used the within branch condition, and they tried to test giving the pulses at different intervals, starting from 0 and going up to 50 milliseconds apart. And you can see that when they're given at the same time and after a short duration, a short interval between them, we have a very large superlinearity. In blue circles are what was actually measured, and in black squares is the arithmetic sum, what we would expect if it was linear. And you can see that there is this nice superlinearity, which decays as the time interval grows, but still exists, until finally at about 50 milliseconds, it's no longer there. It's again as if it was two different compartments and not from the same one. Okay, so now, to summarize. What they tried and convinced us is that the results here indicate a two-layer sum of subunits model. What they claim to show is that there is local sigmoidal summation, this is the within compartment kind of summation, and linear summation of different compartments at the soma. And I want to show you how they try and relate it to other models that were available. So this is basically what I just told you from, I think at least Mel is the same, is one of the authors of this article as well. You can see here these sigmoidal small circles are the subunits, the different compartments, each getting inputs from different parts of the compartment. And then it is summed up. For some reason it's sigmoidal, but they talk about linear summation here, plus the possibility of maybe giving different weights to these subunits. So this is the two-layer model. However, there's another model which is very popular, and it's a two-compartment model, which talks about a distal compartment and a proximal compartment. And this is quite useful in describing things such as back-propagating action potentials, which we talked about with Idan, because the distal part is considered as the slow with the larger time constant, while the proximal is considered as the fast. So it is kind of an important model, and it seems at first, I'm about to finish, it seems that...
5 more minutes, take it.
Great. Okay. So let me talk slow. So it seems as if these two are not really, they can't, it's a problem if we present it this way. And what they are saying is that though they did not directly test it, it is compatible with a three-layer model. The three-layer model basically says, take the distal compartment and replace it with a two-layer network. Take the proximal, replace it with another two-layer model. And then at the summa, we have some kind of multiplication of the outputs of these two mechanisms. This is like a distal integration area, and this is a proximal integration area, and their outputs can be somehow integrated. Of course, this is not something that this article actually shows, as the only thing they did is stimulate a pair at a time. So they had either a pair that was like from here, and they just managed to show it in comparison to what happens if, for example, I stimulate here and here. So they don't say, hey, we proved this, but they say it is compatible with this kind of model. And finally, what they're claiming is subtle compartmentalization, which is a really hard word. And that means that, like I try and show you, they're not talking about a compartment which is really strict and you can just put it nicely in your model, but rather of a kind of a sliding window which has these smudgy edges which change according to stimulus intensity, to the temporal things that I showed you, and so it makes stuff more complicated if you're trying to model it, I would guess. And maybe just to sum up what they didn't test, so it's really important to remember a couple of things. First of all, this was in vitro, in a slice. They only stimulated a pair at a time. It's nothing like you would expect like from in real life. They use sub-threshold intensity simulations, so it's not like the action potential and stuff like that is not really what they're trying to talk about here. And again, this was all excitatory, and they did not. They did test some stuff about inhibitory influences, which you can ask me about, but that's not their main agenda.
Question.
Can you state again, just for us,
Mhm.
what are the two layers in the model? What are the two layers? Which is the first, what's the second?
Okay, so what they're claiming is basically this kind of model. They're saying that
Like, the,
what are the two layers? Exactly. So over here is for example, this is a compartment which has like a certain length of the dendrite in which it has this sigmoidal function. So if you get the really small input, it would be more linear. If that's a medium, it would be more superlinear. And this is basically the calculation done at the stoma, which adds up different compartments.
so why are these two? Which one is the first? Which one is the second?
This is the first,
Okay.
and this is the second.
Adi?
Yeah.
Did they show anything about the inhibitory synapse between A and B?
In between A and B?
A and B.
What do you
Like
mean?
the location A and location B
Yeah.
on the same dendrite.
Mhm.
What if there is some kind of inhibitory synapse between that?
Okay.
So, because they are saying that the nonlinearity is because of NMDR receptor only.
Mhm.
What would
Okay.
be the result if there is some kind of inhibition it will be there?
Okay. So what I didn't tell you is that over here, I don't remember exactly which color it was, but they tested, they put in the extracellular fluid, they put different concentrations of GABA antagonists, meaning that they inhibited the inhibition. And they show that even if when they do not inhibit it, or when they inhibit it like with really high concentrations of the GABA inhibitor, we still get this sigmoidal shape. It does influence the threshold of exactly when, but it's still there. So they did test it.
I'm not sure I understood the sublinear explanation. How did they explain this?
How do they? Well, I think they're not really getting in this article into the biological meaning of what's going on, but I think they're trying to claim that this has something to do with the initiation of action potentials in the dendrite, which inhibit it from, I think it's inhibited from passing it on to the soma, some kind of process like that. Because they tell that what they see at the soma, the kind of voltage traces that they get there, look like it was, there was an initiation of it or something like that, but they do not really explain it. So, yeah.
Is there anything about the relationship between the distance of where you stimulate and whether or not you get a spike at the soma? Like not between the stimulations, but if you're really far away from the soma, and even if you get a spike in the dendrite, will you also get a spike in the soma?
What they did here is most of the time they use small stimulations. They're not talking about action potentials, and as you can see in the soma, it's like, see over here, the scale is 3 millivolts, so they're not talking about action potentials.
Uh-huh.
They're talking about sub-threshold that their entire
Okay,
case.
but even these sub-threshold things, so does it matter if you stimulate far away or if you stimulate closer to the soma?
All the simulations they gave were about 100 microns away from the soma. So I don't think
they're
think
close.
this study is a study which compares the distance. It's not. You can't answer that question with this study. tons of input from the same place, maybe it doesn't give you too much information, so you don't want to act upon it.
Seems like that's important. If you get a lot of information, you wanna
Yeah, but
continue.
I think with tons of information all the time, that means maybe something is wrong there, and that's like one option. And for the superlinearity, I think that due to the fact that they also use the paired stimulation, over here it's just paired, it's not like simultaneously. Maybe it has something to do like with learning and with like coincidence detection or association. So you want to, I don't know, maybe. So that was the last question. Right?
Okay, we take 15-minute break.
I don't know. I never heard about it really.
Yeah,
Associated.
I must
יודעת? שקע פשוט, יודעת?
שקע?
כן.
כן, למטה, מתחת לזה.
יש לו את זה, אבל בשקע.
לא,
נכון, הוא לא
איפה
יודע
שאנחנו
מה הוא.
שוקעת תעבוד
הוא
מולו.
לא יודע.
אותו דבר.
תסתכל למטה.
אהה.
למה?
אה?
כי P1 שלו שווה מה שאני לקחתי במשוואה
לא, אתה אומר שאתה אמור לעשות אותו דבר מצד אחד. יכול להיות, כאילו, אבל לא, זה מה שהוא אמר שבגלל שיש נקודות אינטראקציה קבוע, ואז
כן,
אתה חייב להשתמש
אבל
בזה.
הגבול של P1 הוא בעצם מייצג C סוף P1. הגבול של P1 זה בעצם P1. ו-P2 הוא נעלם, אני לא יודע, P2 כלום לא יכול להיות. זה נשמע לי...
לא, גם כאילו יכול להיות גם P1, כאילו גם אני אבל, אתה מבין, זה P כזה,
לא נראה לי שזה הבדל משמעותי, כאילו, כי אני לא חושב שיותר מדי גדול כזה זה גדול שווה או
רגע,
קטן
עדי,
שווה.
אפשר שאלה? עדי, אפשר שאלה? תגידי, סיימנו את הדיון הזה? אוקיי, מה עשית בשאלה הזאת? והכי
No,
חשוב?
the other problem is too much for me. I mean, too long,
לא,
and
אני רוצה שאלה.
I'm very busy. So I was trying to applicate to like parallel processing, but
לא,
I
אבל
don't know.
הפרקטיקה שלך קבוע, כמובן הכי קבוע.
What? Here? No, I took master.
מה שאתה עושה.
Parallel.
כן, כאילו,
Yes,
בכל
yes.
מקרה יש פה איזה שמכפילים
זה
בעיר,
כמו
משהו.
של האינטגרלים
Getting,
שדנה עשתה.
I like that.
והפריד
About this process and about interpersonal
כן,
process,
נכון, שזה
the
אי...
area between the two
כן, אני עשיתי את זה.
כן, אבל האינטגרלים זה לא הבעיה, השאלה
here you just
השאלה שלו...
get to know the processor and what is going to occur in the here, and you can translate variables and in something like
לא הבנתי מה. אבל
איך מפעילים את זה? מישהו יודע?
הון.
ההון לא... לא עושה מה שציפיתי שיעשה.
לא עובד?
ל-PGA? לא עם ה-PGA? אולי נכנס פה משהו. אני חושב שהגעה, לא קצת חומרות יותר פשוטות מזה, אה? הנה, יש משהו קורה. Okay. So I'm going to present the paper, subcellular mapping of dendritic activity in optic flow processing neurons. So let's go through that long name. So the subcellular mapping means that we map the response to certain stimuli in the subcellular level, means that within one neuron of dendritic activity, means there are units of interest are dendrites. In optic flow processing neurons means in the visual system, to put it in simple words. So we're gonna map the response of dendrites within a single neuron, a single visual neuron. And our animal, the animal that they studied is the fly. So levels of topographic mapping. So we have all kinds of topographic mapping. You can map through the routine topic visual field. connect to different units on some level. And they're responsive to specific patterns of global motion. And when I say global motion, this means, in this context, self-motion. Because global motion is when the entire field of view is moving, and if the entire field of view is moving, it probably means that you are moving. And due to the nature of the motion of the fly, it is mostly rotational. Okay, these are the proposed roles of the VS cells so far. We're going to talk about this kind of blowfly, which is a kind of fly. So this fly has 10 of these VS cells on each side. It's also bilateral like us. So we have 10 of these cells, which is a very small number, which is convenient. And it is located in a place called the lobula plate. So this is how the visual system of the fly looks like, more or less. You have several layers. You have initially the photoreceptors, the retina. Then you have a layer called the lamina, a layer called the medulla. And the final layer in the visual system, the one that sends, it sends neurons eventually to the central processing or already to the motor places. So this is called the lobula plate, and this is what marked with red over there. So if you look with a finer resolution, so again, this is the retina, the second layer is the lamina, the next layer is the medulla, and we are interested in the lobula plate, which looks like this, in this shape. These cells are the VS cells. Okay, they are like T-shaped cells that have their T part in the lobula plate and send their exons to the higher places or the motor places. The fact that there are only 10 of them in the lobula plate doesn't mean that there aren't any other cells. So there are many other cells in the lobula plate, but characteristically only 10 VS cells.
Each layer is a different cell, so this is like 3 synapses away?
Not necessarily 3 synapses away. Because each layer may have vertical synapses between them, and there are several types of layer which that are interconnected with each other. So I wouldn't confidently say that there are only 3 synapses between the retina and
The minimum of 3 synapses, or you also get input from the lamina to the lobula?
I actually can't answer with certainty about what happens in the previous nerves because they don't focus about it in this paper.
Okay.
Are those T cells the only connection between the eye and the brain?
No.
Okay.
They're not the only connection, but they are related with the self-motion. They are the ones which are related with the self-motion. To get back to this slide, the dendritic spanning fields and receptive fields are highly individual. So what am I talking about? Spanning field is the way that the dendrites are organized in the cell. I mean, the density of the dendrites and the location and the length of the dendritic tree itself. It's quite individual between different individuals, and also the receptive fields of the dendrites is not as fixed like with the C. elegans, which is it's very similar between different individuals. In the fly, it's already more heterogeneous between individuals. Yeah.
Individuals mean different flies?
Yeah, different flies.
So you're going to tell us about some VS cells, and you're going to talk about specific instance of specific flies, and different flies would have different characteristics?
So yeah, so they had several flies in the experiment. They're going to talk about only the general phenomena that they saw in all flies, but if you go into specific, like the specific spines and the specific branchlets, so this would be different between flies. But the general behavior of the mapping is similar between individuals.
So what do you mean by highly individual?
A highly individual means that the tree itself look completely different density-wise between different individuals, but the encoding is quite similar, which we'll see.
Different morphology, but similar encoding?
Also similar morphology because they're all T-shaped, but it's not completely the same. Okay. So they're gonna look. I think this is the justification to looking only at average phenomena and not going into specific in the experiments. Okay. They are numbered 1 to 10, distal to proximal, and when I say distal, I mean
Wait.
close, yeah.
Sorry to interfere again about
Sure.
highly individual. Normally, the neurons between VS cells between different flies should be very similar to each other in terms of the
Yeah,
morphology.
yeah.
So these ones are specifically variable, or this is the general?
So no, generally the dendritic span field in flies, as
Very
far as I
similar
understood,
between different flies.
it's not, it's individual. If I look at, choose like it would be easier to explain actually when I have pictures
You
and
put a
graphs.
gun into my head and tell me, is this a VS neuron or not?
Yeah, it's
Very easily
yeah, recognized.
So this is just the height. I'm just wondering, why did you put this word there? Because normally I would think the other way around, that they are generally, I mean, if you look at the final details, yeah, they're different as a...
So soon I will talk, I will mention this again and talk about what I thought they meant by individual. But this is just one sentence from the paper which they didn't further explain. But I'll give my interpretation when I have a graph in front of me soon. Okay, not this one though. So this is again the lobular plate. So you have these are only 2 to 9. So 2, the red one, which means the distal. The distal, when I mean say distal, I mean distant from the center of the animal, from the head. So they mark the most distal one as 1, and then 2 and 3 up to the most proximal one, which would be 10. So this is how this is the way we would name them from now on. So the inputs that these cells get, first of all, they get feed-forward local. Feed-forward means that from the earlier, they get input from the earlier layers. Local means that there isn't a lot of crossing between dendrites. Means that mostly the input to a single dendrite, for example, in the cell will be from close by neurons and not from very distant neurons in the previous layer. Direction selective means that they mostly respond to a specific directional stimuli, like we saw with the cats, with the Yugal and the Weasel. The inputs are from bushy T cells, which is a kind of cell in the earlier layers. So this is the feed-forward input that they get. In addition, they have 3 types of lateral inputs. They have dendrodendritic gap junctions, which is, well, we know what gap junctions is. It's like electrical synapse between dendrites of them and other horizontal motion to neurons. They have proximal and distant VS cells. They inhibit each other through inhibitory interneurons. Means that their computation, the computation that they do actually affect each other through this distant and proximal inhibitory neurons. And they are connected to each other through electrical synapses at their axons. Means that after the soma did the computation with all the inputs from the dendrites, even after the computation is done and I don't know, a rate of action potential started to appear, even after it, they have synapses at the axon level that affect each other's inputs. Yeah.
I just want to verify, these 10 cells get all the inputs, all the visual inputs. It's like a bottleneck.
No, there are, as I said, there are many other cells. These get, I don't know, I wouldn't say it's 1-to-1 at all, but they get some of the inputs and they give information presumably about the cell's motion. So it's not a bottleneck. There are many other cells that the optics flow goes through, but this is one type that they focus about.
What's the difference between VS2 and VS7, besides its location in the lobule?
So for now it's just their location, but we're going to individualize them when we have data. So I'm going to get to data soon. Just nobody here knows anything about the fly, so I felt it's important to give some background. Yeah.
Yeah, a question about the fly.
Yeah.
The dendrodendritic gap junctions and the axons connected to another axon. Now these type of things are characteristic to, I don't know, temporal animals with
So we know we have them in our brain. As far as I know, we have in different places in our brain we have this kind of stuff. But you can go back to this little fly, and you already have this kind of mechanism. I think this signifies the importance of these mechanisms. That even in much simpler neural systems, they take place in computation. Apparently, otherwise, I guess they wouldn't be there. But we have them, and memos have them, and slides have them too.
We have them a lot, because I don't know if you know.
I don't know. I don't know. We have them.
Conjunctions are very common. They're not dendritic led, but you also have them.
In the axoxonic.
Axoxonic, I don't know what is the big data is of axoxonic, but certainly there are. But normally it's not as common. In the single digits, I would say.
So the big question, not the one that this paper proposes to answer, but the big question is how do these VS cells encode their receptive fields through this intricate network? We're going to talk about the receptive fields too. So the question is how do they encode, but the contribution of this experiment to this question is just to examine the preferred location and preferred direction, okay, of local motion cues for defined branchlets across the entire dendritic field of a set of the VS cells. So mostly the contribution of this paper is not proposing a computational model to how these cells integrate the information that they receive into a specific output, but just to give a lot of data about how these inputs look and how these outputs look. So what we essentially are going to do, we're going to look at like these branchlets in most of these VS neurons. At each branchlets, we're going to find out what its preferred field in the retinotopic view and what its preferred direction of movement. Okay, this is the basic experiment. So data and stimuli. So first of all, what they did is calcium imaging with the indicator OGB1. With relation to calcium, insects have nicotinic receptors which are found on these branchlets and are permeable to calcium. Means that we can see signals of excitatory inputs because when there is a synapse or when there is a channel opening in one of these branchlets, calcium also goes in, so we can see actual individual inputs of individual branchlets in the cell. And also they have voltage-gated calcium channels, which means that we can see the eventual membrane potential also using calcium. So calcium imaging is I guess it's a legitimate method for studying these neurons or insects in general, as far as I think. But I would stay specific at these neurons in this type of fly. So you can see also the excitatory input and the eventual membrane potential with the separation, and we'd be able to separate them. The stimulus is an array of light emitting diodes. So there's many diodes, not just 5 over 10. These are the bins that they're going to use for the stimuli, but there are many LEDs over there. This one is presented in front of a single eye. We measure the position of the field with the parameters azimuth and elevation. Their stimulus spans only 180 degrees of azimuth and 90 degrees of elevation, which is most but not all of the visual fields of the fly. So they're going to talk about only the cells numbered from 2 to 9, which is the second to one before last of the VS cells, because the other cells visual field goes out of this visual field of the 180 over 90. Okay, so they didn't include their data. Okay, so this is how the stimulus look like. We have this 5 over 10 bins. Again, each bin is comprised of many LEDs. So what they did, for 5 different elevations and 10 different azimuths, they presented a moving bar. Okay, either to the right or to the left, or in this case, down or up. And for each one of the bins in their simulated visual field, they determined their preferred direction of movement. So for example, if we look at this, so for this specific bin of the visual field, this is the calcium response for a down movement, and this is the calcium response for an upward movement. Let's not talk about C, D, F, and G yet. Okay? And for example, here you can see that for a left movement, you have this kind of response, and for a right movement, you have hardly any response at all.
Rick,
Yeah.
they're showing a couple of, like multiple bins simultaneously, or just one bin at a time?
It's a moving bar. So they can check
Okay.
when this bar moves along this row, they can check each one of the beans in this row.
Okay. It's individual.
Yeah.
Okay.
So each bean will get its individual preferred direction.
How is it possible to do such visual experiments with light? If light is fixed, how can they
Yeah, it's anesthetized, its legs are cut, it's fixed, whatever you need for it to work.
Yeah, it's not anesthetized, but
Not anesthetized? Ah, just
you don't need to anesthetize, ethically, you don't need to anesthetize insects.
want to cut the legs and
Cut the legs, etc., and put them on.
Okay, so no, no need for anesthesia. Okay, so for each one of these beams, which represent a part of the visual field, we get a response for down, up, right and left movement. So let's talk about this further graph. So first of all, they saw with a very brief stimuli, they saw that the response of the calcium indicator, OGB1, is very fast going up and pretty slow going down, which is something we are already familiar with. So in order to not give any beans too much preferred direction that they don't really encode, they did a deconvolution of this shape from the shapes that they got from the calcium imaging to get a sharper, more symmetrical responses. Deconvolution means that assuming that a very thin response was smeared to the right, then deconvolve it in order to make it look like a composition of three thin responses without this slow decay. So C and F would be the deconvolved graph, and D and G would be the binned version of C and F. Means that I don't need this continuous, like probably hundreds and thousands of points. I only need 10 points for azimuth, so I average over each one of these bins to get its very specific response.
So Omri, this is response from all of the neuron or part of the dendrite?
So for each dendrite on each BS cell.
Yeah.
What?
These responses, what is the region of interest of
Different branches in different BS cells.
so this is just random example.
So yeah, so this is a single branch, okay, out of many. So the next part after, for each beam they could write a response for each one of the four directions. They could do a vector summation. I don't know if I agree with this method, but this is what they do. They did a vector summation, which means that they sum up the blue and the yellow, which is up and down, and they summed the green and the red, and they got a summation. This is what they call the preferred direction of these specific branches. Okay, so this means that if this branchlet responds completely the same for up and down movement, then you would get nothing on this dimension. This is why I'm not sure this analysis is the right way, but anyway, this is what they've done. Wow, 5
minutes.
Okay, so they did some controls. They did square grading also in 8 directions, and they got the results are not significantly different. So if anyone is going to ask about the amount of direction or the shape of the stimulus, they did a control for it. So this is one of the VSLs. This is VS4 in one specific animal, and you can see the visual fields of each one of 13 branchlets, which is not they checked many more, they just presented 13 here, especially for VS4. So what they did here, they smeared the LED array for it to match the density and the shape of the fly's eye, okay, already in the picture. And for each branchlet, for each beam, they marked an arrow that symbolized the preferred direction of this branchlet to this beam of visual field.
The eye you showed before, like
So
the
this,
summation of the vectors?
So yeah, this is the vector. The arrow is the vector summation for this specific beam on this specific branchlet in this specific VS cell. So the correlations that they found, the correlations was prevalent in all the cells they checked. First of all, when you go from dorsal to ventral, you can see that the preferred visual field goes down. Second, when you go from dorsal to ventral, also the preferred visual field goes from pretty much horizontal to pretty much vertical. And also there's a correlation which is not as visible in this example, but there's also a correlation with the azimuth position. So they found a significant correlation with azimuth, with elevation, and with direction along the dorsal to ventral route in the dendritic tree. So this already start to resemble like sort of a rotational movement of the general local cue. Okay, afterwards, they took all the branchlets from this is one specific animal, they took 270 branchlets from all the 8 VF cells that they checked and tried to make a more population or more general mapping of the lobular plate. First of all, to check if there is a correlation between proximal, close by, nearby branchlets, they did a pairwise correlation between all of these 270 branchlets and saw that it's more or less along a straight line. Means that there is a pairwise correlation between branchlets in this lobular plate in general. And then they just did an azimuth map, an elevation map, so each color encodes different azimuths, and saw that there is a gradual mapping along the branchlets, both in the azimuth and in the elevation dimension. And they did a color map that more or less symbolized the same thing. Okay, so this is pretty much the last figure. So now they, with the same method, they checked the preferred direction and visual field of the output of the axon, and compared a summation, a linear summation, unfortunately, but they said it's just as good as passive cable theory or whatever. I'm not completely convinced, but okay, they did a linear summation of all the dendrites in the dendritic tree and defined the general visual field of a specific VS neuron. So this is VS2, VS3, and so on, and compare it to the preferred direction for each beam of the axonal of the axon going up. And we can see that the visual fields are pretty much look alike on some level, but there are fine differences between them. Mostly it seems like the axonal what? Yeah. The axonal field is more smooth than the dendritic summation. So they gave a reason, which to me makes a lot of sense, but does anyone ever guess why the summation of the dendrites along the dendritic tree and the eventual output of the axon, why the field doesn't look exactly alike, but only partially alike? So it's something I said at the beginning of the presentation. The reason they gave, and it makes actually a lot of sense to me, that there's after the summation of in the soma, there's a summation of a lot of inputs, and then an action potential is created. After it, there'll still get electrical synapses between the axons of the different cells, which can still change each other's output, eventual output, mainly by smoothing it, and it does seem much smoother in the axon case. So this is the way to show the connection between the sum of the dendritic inputs to the eventual axonal output. So to summarize, they found retinotopic mapping both on population and subcellular level in the lobular plate of the blowfly. Both correlates with rotational self-movement, as we saw. It makes sense, though they didn't offer a computational model, just presented data. And the similarity of input summation and output, which is the last figure we saw, similarity between them.
I have a question.
Yeah.
Can you go back to the slide with the VS4, the big one? Yeah, this one. Or you told us there's a correlation between the preferred receptive field and 3 different things. So what do you correlate? What do you mean by preferred receptive field, and what do you correlate to what?
Okay. So the correlation is between dorsal to ventral axis, okay, to 3 different things. One is the azimuth, which is the location, the horizontal location of the lead, I guess. So they found the correlation. I didn't present the numbers. They did some statistics. They found the correlations between the dorsal to ventral position of the dendrite and the pin with the biggest reaction, okay, the biggest response. So you take the pin with the biggest response. This is the most preferred field or preferred position of this specific branchlet.
So which one? This way.
So for example, these branches, this would be.
And how
so this would be the azimuth,
Okay.
and this would be the elevation. So each beam has its coordinates of azimuth and.
So you pick one beam.
So I take the beam with the strongest response, and there is a correlation between the strongest response beam and the dorsal to ventral position of the branchlet, both on the azimuth and on the elevation axis separately. And there's also a correlation between the dorsal to ventral position of the branchlet and the direction of the most responsive beam. So there are three types of correlation. This is probably a best figure to give this example of, but they say they found this correlation in many animals and